Here you go. Everything inside here. You're all good. Feeling good about it? You just let yourself Great job.
We try to get
Good morning, everybody. If you want to come in and find a seat, feel free to grab some coffee, pastries in the back, and get up as needed throughout the presentation to help yourself to refills and refreshments. We're going to get started in just a minute here. There's a few more seats up front, and I know seats in the back. We'll have some people joining us over the next few minutes as we get started here, so just make sure you make it obvious that there's some seats next to you. We may have a full house this morning.
I work in the building, so it is. I'm actually able to, I live on Mendota Bay, so I can walk or bike back to work. St. Goods is Johnson's. It'll be interesting to see how this sorts of things.
All right. Well, welcome everyone to Yahara Lakes 101. My name is Elizabeth Kottreinders. I'm the Director of Policy and Communications here at Clean Lakes Alliance. And as always, we're happy to see a full house for our Yahara Lakes 101. It's great to see the usual suspects who regularly support this series every month, as well as brand new faces every time. So we're happy to have you here. We want to thank some people before we get started. So Yahara Lakes 101 is co-produced by the Clean Lakes Alliance and UW-Madison Nelson Institute for Environmental Studies. So we want to thank them for their partnership. And we want to give a very big thank you to Foley & Lardner, the law firm that occupies most of this building. They are the hosting sponsor of Yahara Lakes 101. And not only do they provide the space for us to meet here, they very generously provide a suite of offices for the Clean Lakes Alliance on the second floor of this building. So we would not be here without the generous support of Foley, so we want to give them a very big thank you for their continued support that way. I just have a couple of announcements before I introduce our speaker today. So if you pick up your table tent and look, we've got a great lineup for the rest of the fall for Yahara Lakes 101. We do not meet in December, but you can see next month we have Dr. Chin Wu, then we've got Dr. Ken Bradbury, and then in November we'll actually have a panel of Clean Lakes Alliance staff that will be talking about our citizen water quality monitoring program and some exciting initiatives and programs that have come out of that. So we hope to see you for those. I want to encourage all of you, if you're not yet, to become a friend of Clean Lakes Alliance. It's a minimum of $35 donation annually, and what that does is it gets you free admission to Yahara Lakes 101 each month. Some of you you're here for your first time, pay $10 to come. We want to thank you for your support of this program and the work of the Clean Lakes Alliance. For $25 more, you can come each month to these without paying each time. There's lots of other perks, free information, education, and early access to get tickets for some of our big events that sell out. So there's people at the registration table that can answer more questions about that, but please consider becoming a friend. And another thing I want to mention is that depending on what happens with the construction at the Edgewater and how soon that happens, we may or may not be moving the venue of Yahara Lakes 101 over to the Edgewater within the next couple of months, which will provide a little more space and possibly an easier parking situation. So stay tuned for information on that. In the meantime, plan on coming back here for the next month, but we will keep you posted. So with that, I'm happy and honored to introduce our speaker, Dr. Trina Mcmahon. She's a professor at UW-Madison with joint appointments in civil and environmental engineering and bacteriology. She's been studying the Madison Lakes since she arrived in Madison in 2003 after getting her PhD at the University of California at Berkeley. And she's also currently a member of the Dane County Lakes and Watershed Commission. So we're very excited to have Trina here. We have a great presentation ahead. We're going to let you all know whether she wants questions throughout or hold them at the end. We'll see how this goes. If there's ever something you don't understand, just put your hand up and we'll try to get that clarified as we go. This is a pretty casual event, so feel free to chime in with your questions, and she'll let you know if she wants to hold or keep them coming throughout. So with that, will you please join me in welcoming Dr. Trina Mcmahon.
So one thing Elizabeth didn't mention is that we're on camera. So I think this is being recorded so that it can be watched later, but also so those who weren't able to attend today can watch it remotely. So that's no pressure or anything like that. Press is going to pick up on this. I was saying it's kind of cheater to put the word toxin in your title because that really draws a crowd. So thanks very much for the invitation. This is a really neat series, and it's really fun to be able to come and see you all. I want to not be too much in the professor role here. I don't, I do teach at UW, and I teach all from freshmen all the way up to graduate students. But I don't want anyone to be intimidated by that. I am also just a citizen of Madison. I sit on the Dane County Lakes and Watershed Commission as a citizen of Madison who cares about the lakes. I happen to do some research that relates to cyanobacteria, and that's what I'm going to talk about today. But certainly, please raise your hands often and ask lots of questions throughout. I might call on you, or at least ask for volunteers from the audience to help me out a little bit as we go through our talk today. And so as Elizabeth mentioned, I have a dual appointment. I'm in engineering and in bacteriology. And I'm trained as an engineer, and I'll tell you a bit about my background so that you know where I'm coming from in terms of my expertise and sort of the limits of what I do and don't know. But I want to talk today about Lake Mendota mainly, but of course a lot of what we learn about Lake Mendota applies to the whole chain of lakes in the Yahara chain. And I'm going to talk a little bit about nitrogen, which is something maybe you haven't heard about, because I bet you all know why our lakes are green, but I will come back to that. So this is just a sort of big picture overview of my research group, and this is, I guess, encapsulates the entire water cycle as I view it. So I was trained as a wastewater engineer, so I actually studied the removal of phosphorus from wastewater when I was a graduate student, and so we continue to do that in collaboration with the Nice Springs Wastewater Treatment Plant here in Madison. But I'm not going to talk about any of that work today. Most of what my current research grant dollars are going towards is studying the lakes. And so we study mostly bacteria that live in the lakes, not just cyanobacteria, but also the, I would say, healthy, natural flora of bacteria that live to the order of 1 million cells in a drop of water. And those are not bacteria that make you sick, they're not E. coli, they're not anything that would ever infect you. In fact, they are in fresh water all around the world, the same kinds of organisms. So that's mostly what I study in my lab. I've had a couple of people move through my lab, graduate students and a postdoc, who are very interested in cyanobacteria. So most of the work I'm going to show today relates to cyanobacteria. And those two people are Todd Miller and Lucas Beversdorf, so I want to make sure that I give them full credit. Todd is now at UW-Milwaukee in the School of Public Health. So he doesn't study the Yahara Lakes anymore exactly, but he is extremely active in the area of cyanobacteria research, so if you wanted to follow up on any of that, you should go check out his website. You can find him easily by Googling UW-Milwaukee Todd Miller. Lucas graduated from my lab with a PhD last year, and one thing you'll notice in the data that I'm going to show you today, and the results that I'm going to show you, is that it goes in 3-year increments. Now, who knows what goes in 3-year increments at the university? Any ideas? PhD program. So Lucas did a PhD. He was there for more like 4.5 years, but he had to ramp up and ramp down. So we got 3 years of data, really good data from when Lucas was there. Then we had a gap, and now I have a new graduate student who's picking up on the work that he was doing. So most of what you're going to see are coming from Lucas's PhD. And he's now at UW-Milwaukee working with Todd, so he's continuing his work on cyanodes as well. So why are our lakes green? Does anybody know? Phosphorus. Okay, so I'm not going to talk about phosphorus. You're all like, oh man. Right up front, I'm going to say our lakes are green because of phosphorus, and so I'm not arguing that. I think people hear our work on nitrogen and assume that we're arguing that we shouldn't be paying attention to phosphorus, but I think we absolutely must be paying attention to phosphorus. So all the work that the Clean Lakes Alliance is doing and other groups to try to get phosphorus out of the watershed, to keep it out of the lakes, absolutely needs to be done. I think the work that we're doing on nitrogen tells us about what happens in a lake within a particular growing season. So from when ice comes off to when ice goes on. Nitrogen becomes very important during those time scales, not so much over years to decades like phosphorus is. So you've got to tackle the phosphorus problem, but you also need to understand nitrogen if you're going to try to predict when and if these blue-green algae are going to bloom or cause toxins to accumulate in the water. So I just want to get that right off the table, right on the table, because I don't want to shortchange phosphorus, even though I'm not going to talk about it very much. So cyanobacteria. Let's talk about cyanobacteria. These are cyanobacteria that come from Lake Mendota. So these were images collected by Lucas Biebersdorf. And people who study cyanobacteria for a living love them because they're beautiful. When you look at them under the microscope, they have all kinds of interesting shapes, and they store things inside their cells that make them, under a microscope, have all kinds of beautiful colors, especially if you stain them chemically. And these are some of the usual suspects that we find in Lake Mendota. They are particularly beautiful. You see a Phycocyanin is up in the upper left, and I didn't put the names on here because it's not really important, but if you're a cyanobacterial junkie, and there might be people in the audience. We also have Anabina and Microcystis, and more recently, we see a resurgence of Gliotrichia. And so I'll talk a bit about that later as well. But so these are kind of the usual suspects looked at under the microscope. When you see them in the lake, it mostly just looks green, right? This bluish green color. One other point I want to make, and this is the bacteriologist in me, is that blue-green algae is actually a misnomer. So blue-green algae are not algae, and this becomes important when you think about trying to remove them from the lake or use some kind of treatment method to get them out of the water if you're going to use it for drinking purposes. Cyanobacteria are much smaller than true algae, and the misnomer blue-green algae came when phytoplankton ecologists, so people that study phytoplankton, the green stuff in lakes, started looking at these organisms, hundreds of years ago. They looked like algae because they were green. So they said, oh, they must be algae, but they're a little bluish, so we're going to call them blue-green algae. But then when scientists look deeper at their DNA, we see that they are much more similar to bacteria than they are to algae. Okay? So they're more similar to E. coli than they are to a plant. Right? But operationally, for the purposes of communicating with the public and making policy on how to keep them out of the lakes, probably doesn't matter because they use phosphorus and in the same way algae would, and they are green, which makes people upset because the water is green. They are unique in the sense that they make toxins, and so that's largely what I'm going to talk about today. The algae, the true algae that grow in lakes, do not tend to make a lot of toxins. It's the cyanobacteria that are responsible for the toxins. And that's different than what you find in the ocean. I'll say a bit more about that. So my title mentions nitrogen. Now, I also didn't want to short shrift phosphorus, but does anybody know what this represents? Or can you guess what this might represent? It's a chemical formula, sorry. I don't show very many. Huh? You can't? Any guesses? Don't be shy. Huh? It's a carbon chain. It represents a molecule. I mean, it's actually like kind of an average composition of a molecule. It's actually the average composition of a cyanobacterial cell, right? So they're not any different from you, really. when you get down to it. They need carbon to build their cells, they need nitrogen, they need phosphorus, they need a lot of other stuff. Okay? And this list goes on. You need cobalt and nickel and all the trace elements that you find in a vitamin pill that you take, right? So they build their cells the same way, essentially, that you do from from the fundamental elements. Now, the reason why we are so worried about phosphorus is because these are in order of their stoichiometry, the order in which they're important for the cell. Okay? So they need a lot of carbon. They can get carbon from the atmosphere because they're like plants. They take CO2 and they make it into their cells, right? So that's why they have pigments like chlorophyll to capture that sunlight. Nitrogen is next, right? So they need a lot of nitrogen. It goes in their proteins, and you have proteins, so they need proteins. It goes in other components of the cell. It's actually a component of the toxins. The toxins themselves look a little bit like proteins. And then comes phosphorus. So if they don't have phosphorus, they can't build their cells. But they also need nitrogen. And of course they need carbon, but they can get carbon from the atmosphere. The thing about cyanobacteria that makes them really different from algae and from many other organisms in the lake is that they can also get the nitrogen from the atmosphere. So they can take nitrogen, dinitrogen gas, N2, which is normally a pretty inert compound, and turn it into their cells. And that is something very unique to bacteria. And it also means that they can, eventually that nitrogen can end up back in the atmosphere because it gets cycled around by when the cyanobacteria die, it gets recycled by other bacteria and sent back into the atmosphere as dinitrogen gas. That does not happen with phosphorus. Right? Phosphorus, there's gaseous forms of phosphorus, but they're really toxic, and thank goodness they don't accumulate to any measurable amount in ecosystems. But it means that the phosphorus stays in the lake, whereas the nitrogen cycles around into the atmosphere and back in. And so the reason we focus so much on phosphorus when we're trying to control cyanobacterial biomass in the lake is because once it gets in the lake, it doesn't leave. I mean, it can leave through the Yahara, in terms of Mendota, it can leave through the Yahara River, flowing out when it's tied up in a cyanobacteria. But mostly it doesn't leave. The nitrogen can leave, but it can also get in. So the data I'm going to show you today focus on how cyanobacteria get that nitrogen, because during certain times of the year they have plenty of phosphorus. We know there's a surplus of phosphorus in there. So how much nitrogen they can get that at that point in time completely controls how many of them can grow, and we think how much toxin they can make. So we have to understand that nitrogen cycle. I don't know if there's really anything that we could do to mitigate it, right? So I know everybody here wants to clean up the lake and find a way to clean it up. I think, on the time scale that we care about over the next year to 5 to 10 years, a lot of what we should focus on probably is the work that First Resource does in the public health department, which is just making sure people know when the cyanobacteria are there or not, when they can be producing toxins. So we're focused on when and why they do it. Maybe not so much how to stop them from doing it. Because we know to stop them, we have to get rid of phosphorus. But that's a longer term project that lots of people are. Okay. So this is an analogy that, again, Lucas Bebersdorf came up with. He actually started as an oceanographer, so he took a lot of what he learned in the oceans and brought it here to Madison to try to understand how the lakes function. Which is ironic because it usually goes the other way around. A lot of what we know about aquatic systems comes from studying lakes, especially Lake Mendota. It's one of the most studied lakes in the world, which I'm sure you've heard before. But he brought it the other direction. He brought a perspective that no one here really had before. So in the ocean, there's an organism called Trichodesmium. It's up here on the left. If you remember my pictures of the cyanobacteria that I just showed, it looks remarkably like a cyanobacteria that you would find in the lake. It happens to be kind of reddish color instead of the pigments that it has. And what oceanographers have noticed is that Trichodesmium blooms in parts of the ocean, particularly around Florida and off the west coast in some places. And it blooms in the spring, a lot like our cyanobacteria in the lakes do. So it's the orange squares, rotating squares, and they bloom when the phosphorus concentrations or when the elements that they need are present in the ocean and when it gets warm. And then after they bloom comes something called red tide. So everybody, what's up with red tide? Why do we care about red tide? What is it? So it makes toxins. So they make toxins, right? Just like the cyanobacteria in our lakes. Actually, the red tide is Carinia brevis, which is a true algae. So it's an example of an algae that actually makes toxins. We don't see these organisms in fresh water. But the important thing is that you see this red tide coming after this bloom of trichodesmium. And oceanographers came up with this conceptual model that explains that trichodesmium kind of paves the way for Corynea brevis. It takes a lot of nitrogen out of the atmosphere and puts it into a form that the Corynea brevis can use. So Corynea brevis couldn't grow in the ocean in these parts without that nitrogen being pumped in by trichodesmium. That's the model. That's the conceptual model. And so Lucas knew this, and he said, well, you know, there's probably something similar going on in the Yahara lakes, because, you know, we see these waves of cyanobacterial blooms coming throughout the summer, and there's different species, and there's sort of some understanding of how to predict when those different species will be there, but he wanted to know if nitrogen could explain that, and if that would be a similar mode of action. So, interestingly, the organism that blooms first in Lake Mendota is a phenomenon. It looks remarkably like a trichodesmium, even though they aren't related, very genetically related. They have a similar shape and function in the ecosystem. If you've ever seen a phenomenon in the lake, they're the ones that look like grass clippings. They haven't been around as much over the past 3 years. In 2008, 2009, there were tons of them. But at least that you'd be able to see the eye, because we can find them certainly by the They are always followed by a big bloom of microcystis, which is green tide, right? We don't call it that. That's the organism that we tend to worry the most about. I mean, it's not the only organism, but it's the one that makes us sweet toxins that public health officials worry about human and animal exposure. So this analogy was really what drove Lucas's PhD research and sort of changed how we think about the nitrogen cycle and the succession or the timing of how these different blooms happen, and I would say in the other Yahara as well. So that kind of sets the stage for what we're going to talk about. And if you want to read this in great detail, Lucas published a paper. I'm not going to make you read this abstract or quiz you on it, but if you wanted to go and look at it, you could probably just find it by Googling my name, Lucas's name, and nitrogen. And it's about Lake Mendota. So, but before I get into some of the findings of that, I want to set the stage for what we call harmful algal blooms in freshwater. I told you a bit about it already, but I want to emphasize that this is not a problem that's localized to Madison. It's nationwide. Of course, I'm unlucky that I got the introduction last week of this topic through the press about Toledo, right? So Lake Erie has had problems with cyanobacteria for a long time. I think it's just getting in the press because it's causing shutdown of their drinking water treatment systems. I mean, it's been an ongoing problem. Whether or not it actually causes a drinking water problem, it varies from year to year. But certainly impacts recreation. It impacts other uses of the water besides drinking as well. And there's a lot of really great research being done on Lake Erie through the National NOAA. I don't know if I get that acronym right. At the Great Lakes Environmental Research Lab in Ann Arbor. So if you're interested in that, I would say go to the website because they really stuff there. And we're actually going out on a cruise on Lake Erie in September to look at various aspects of the water quality there as well. So maybe you saw this image in the press. This is at the drinking water intake in Lake Erie for Toledo, and just underscoring that even if this goes through a drinking water treatment system, maybe not something that you want to be drinking. I mean, just viscerally, you have a reaction to that, right? Like even my 4-year-old looks at green water and is like, "Mommy, that's gross." Like even though he doesn't understand what toxins are. I mean, we are hardwired to not want to drink green water. And this is what we saw on Lake Kiganza in 2008. So just when I saw that image from Toledo, I pulled this one out of the depths of my hard drive and we were like, wow, it's really not that much different. So certainly a problem in the United States. You go abroad and there's plenty of problems there as well. I would say Europe continues to struggle with cyanobacteria blooms as well. China, this was Lake Taihu in 2007. And if they continue to have problems, it just happened to be one of the biggest episodes. So they had the drinking water treatment plants in Wuxi were shut down, and it also supplies part of Shanghai, and so it was like millions of people that didn't have drinking water because of the cyanobacteria blooms. And their problems are largely because of untreated sewage, also some agriculture, but Lake Taihu is sort of a China anyway, the archetype for this massive phosphorus pollution that they're struggling with now. So anything that we can learn about how cyanobacteria make a living in lakes and what kinds of toxins they produce has implications for basically global living in terms of water quality. So that's one reason why we study them. Not just because we care about Madison lakes. So I promise not to show too many chemical structures, so this is, I think, the last one. This is microcystin, and I'm going to talk a bit more about it, so I wanted to just put it up here. You don't have to know the details, except note how much nitrogen is in it. Okay, so there's a lot of nitrogen in this molecule. It's actually made by stitching together amino acids, so instead of making a protein, the cyanobacteria make this. Cyanobacteria make a lot of these kinds of things. Okay, so we call them secondary metabolites because they aren't thought to be key to the function of those cyanobacteria. But they make a lot of different ones, and so everybody says, well, they must be doing it for a reason. They've evolved to make these. But actually, we don't know exactly why they make them. There's a lot of competing theories about it. And I don't want to get into it because it involves a lot of molecular biology that I don't really fully understand all the time. Lucas can tell you more about it. But it's basically a molecule that they make intracellular. They keep it inside their cell. It doesn't get secreted. And that's important when you think about drinking water treatment. We don't drink the water from our lakes. We drink groundwater. But if you were going to try to treat this water, or if you're in Toledo, then you would want to think about where that toxin is when the water first comes to your doorstep, right? It's in the cells. So it's not until the cells die, so where the blooms get really scuzzy on the surface and start to break open, that's when the toxin comes out. Okay? When they're happy and healthy in the water column, it's tied up inside their cells. Some interesting health-related facts on this. The mode of action for the toxicity is at the liver, so it's a hepatotoxin. It actually poisons your liver. There's other toxins that are neurotoxins or affect other parts of the body. I'm not going to talk a lot about those. This is the toxin that we look most often for in my lab. We use it kind of as an indicator for all of the toxins. We know that that's not always perfect, but it's the one that track most frequently. And the World Health Organization has recommended limits for how much you could be exposed to. It's about 20 parts per billion, so that's 20 micrograms per liter for exposure through swimming or recreation. If it's 1 microgram per liter or above, then they would put a limit on drinking that water. So that's why Toledo shut down, because they were detecting, Toledo's drinking water treatment plant was detecting microcystin at levels above 1 microgram per liter. Okay. So keep that in mind because I'm going to have some numbers on the following slides. Yes?
So Trina, does microcystin have carbon and phosphorus in its cellular structure?
It has carbon but not phosphorus. So each of in these kinds of chemical structures, the carbon isn't shown, but each one of these is a carbon. So it has lots of carbon. I should have calculated the ratio of carbon to nitrogen in this to see related to the total cell ratios, but it's enriched in nitrogen relative to other things in the cell. So it is enriched in nitrogen. So our whole hypothesis hinges on the idea that the cells are taking nitrogen out of the atmosphere in order to build their cells, but they also build this. Right? So the nitrogen that goes into the toxin is actually coming from the air. Okay, so I just wanted to start with some numbers to give you a sense of the data that we've collected. And I don't want to scare people, and again, I know this is being filmed, so maybe I should be careful. These are numbers generated by Lucas Beversdorf and Todd Miller, so the next few slides are all their work. And we started monitoring for microcystins in Lake Mendota in 2008. So our first data set came from 2008. Anybody remember what was unique about 2008? Yeah, crazy, no, actually opposite. Crazy floods. Crazy floods, right? That was when Lake Delton drained, right? So really abnormal year in terms of rainfall, in terms of what turned out to be the cyanobacterial season. Probably worst cyanobacterial season that we've seen in a long time. We didn't know that going into this. It was just that all of a sudden, well, Todd came to my lab and said, I really want to study cyanobacteria. I said, go for it, go out on the lake. It happened to be one of the worst years in a long time. So we actually saw blooms and toxins that we haven't seen much since. 2009 was also bad, but 2008 was pretty bad. So we did exceed 20 parts per billion, so micrograms per liter is the same as parts per billion. So if you were keeping track of this exposure limit, then you'll see we got above it on one day in Lake Mendota. These are measurements taken out in the middle of the lake. Right? We can talk a bit about this later, about the difference between what's happening in the middle of the lake versus what's happening on a beach or in front of your house, because there's a lot of variation in the, I would call the XY dimension, so across the surface of the lake, and when you go on a transect from here to the north side of the lake, you would find wildly different concentrations or amounts of cyanobacteria. And that's because the water is moving more slowly than the bacteria grow. If that makes sense. Right? So they're growing as the water is moving. It's actually a really complicated physics problem to try to figure out where they go and how they grow while they go. Something that Chin Wu, I see he's talking about Cherokee Marsh, but his work that he does on Lake Mendota, trying to understand how the water circulation patterns control the cyanobacteria, is pretty amazing. So you can come to ask about that. So I guess the point here is that I have day number, which may not be easy to interpret. I have months in subsequent slides, but basically this is July, early July. Early to late, between early and late July is when we usually see the peaks in toxin concentrations. Sometimes in June, late June, depending on when the blooms start. We also often see a peak later in the year, in October, but not very many people are recreating on the lake in October. But presumably people's dogs might be drinking water from the lake as well. So this shows 2009. And this is a slightly different format of the plot, but again, I just wanted to show that the peaks that we saw, there were twice as much as what we saw in 2008. So 2000, across the entire season, 2008 was pretty bad in terms of the density of cyanobacteria, in terms of the intensity of this particular bloom in 2009, it was even higher. And for those who are used to looking at plots, this is a log normal plot, or sorry, a log axis on the y-axis, so this is a tremendous amount of variation. The point being that it's normally quite low. It's usually less than 1, but you have these spikes going up to in excess of almost 50 parts per billion. Yeah.
Is this a residual of the high water in 2008 that you think in 2009 or why?
We don't know, but I can tell you my hypothesis. I think that, 2009 we had a strange spring as well, but a lot of what sets the stage for the summer blooms happens in the spring. Dick Lathrop can tell you about how much phosphorus goes in when you have a big rain event in March when the ground is still frozen and more goes into the waterways. But what I think happens also is that there's an echo. So what happens in a particular summer influences what happens in the fall, what happens under the ice, and that sets the stage for everything in subsequent spring, which then sets the summer. So there's almost like this, it gets dampened as the years go on, which is by 2010, it probably like that. And we don't really know how long that echo lasts. And we don't understand all of the chemistry or the biology that's happening under the ice that would influence that. So it's this combination of what happened in the prior year, along with what happens in the spring that year. Those interactions. So I think that the winter is probably very important. But we go figure, we don't have a lot of data from the winter, right? It's hard to convince graduate students to go out and drill through 5 feet of ice. They much prefer to go out right now. So we don't have a lot of data from the winter. That I would love to get more and to understand better how that's influencing the cyanobacteria in the subsequent summer. So there's a new professor in civil and environmental engineering who looks at this sort of thing. His name is Paul Block. And he looks at forecasting water quality maybe on the time scales of like a year to several years out. So his main work is actually in Ethiopia, forecasting water supplies in their reservoirs. They want to know like 6 months to a year out how much water they're going to have. So he's now starting to look at the topic of forecasting cyanobacteria on that same time scale. So he might have some interesting thoughts. This shows 2010, and I'm sorry, but each of these plots has slightly different format, but basically they were put together from several different places. And what you should focus on is the red. Okay, so red is the toxin, and here we're peaking up at around 12 or 13, so not nearly as high as in 2008 or 2009. What I'm going to talk about later is the green dots. The green dots is the activity of the cyanobacteria related to nitrogen and getting it out of the atmosphere, called nitrogen fixation. So what Lucas was very interested in, which is the subject of that paper that I showed you, the abstract that I showed you, is that you see this surge of nitrogen fixation that happens right before the toxins surge. Okay? We don't have data on nitrogen fixation from 2008 or 2009, which I didn't show it. We really only have it for 2 years because it's a hard thing to measure. So you need to have a very dedicated PhD student to do it. So let's look at 2010. So about the same. Oh wait, this is 2011. This is 2011. And again, peaks in nitrogen fixation right before you see the peaks in toxin. So what I take away from this set of slides, actually here they are all together, that I can make my point. 3 years laid on top of each other, and these are finally now we have months on the x-axis. That's good. So this is really the period that we worry about, so sort of mid to late June through August, and you see a lot of variation. You have different year-to-year variation, but then you have even day-to-day, week-to-week variation. And actually what you'll find is that if you go out in the morning on a Wednesday and measure the toxin, and then go out at noon on Thursday, you can get wildly different numbers if you go to the same spot. And that's because the blooms are growing, dying, moving around, being blown by the wind. So this question of where to measure it and how often really plagues us, because we can't be on the lake all the time. We would love to have an automated monitoring system, which we're talking about. And we would love to have, of course, an army of graduate students to maintain that, but that's a little bit more expensive. And those of you who are familiar with the Center for Limnology probably know John Magnuson, who is also a long-term member of the Lakes and Watershed Commission, and a really amazing researcher and all-around amazing guy. Had the foresight to set up the long-term ecological research program here at Madison. So it's a National Science Foundation funded monitoring program that goes back 25 years now, or almost yeah. I don't know how far back you counted, but the grant has been running for over 20 years. And he coined this term of the invisible present. So the idea being that if you go out during a single year and look at what happens in the lake, you'll learn something about the lake. But guaranteed it's going to be different next year. And it's going to be different the year after that. There's going to be some commonalities, it's always going to be green. But the subtleties of when it's green and what's making it, exactly what species are making it green are going to be different. And so far what I've shown you is data from 3 years. So, well, I showed you 2008, but we really only have really high resolution toxin data for 3 years. 9, 10, 11. And so if you look at that, you see, there's this recurring toxin, and it's actually coincident with the microcystis abundance. So I didn't show those on the previous graphs, but basically, whenever you see toxin, you also see this surge in microcystis because we think that's the organism that's making a lot of the toxin. But we have microcystis data going back much longer, all the way into the 90s because of LTBR, the long-term ecological research. And even before that, because of some bacteriologists that worked here in the 70s and 80s. But we don't have toxin measurements, so we don't know whether it was higher, lower, if it's about the same. So this is a challenge to ecological research, ecosystem research in general, that you only see what you're looking for. So we now have very sophisticated analytical techniques to see this toxin, but the techniques might get even better next year, in which case we might see even more. So this question of how often, how deeply to look, and are we looking in the right place is something to always keep in mind. Because I'm showing you data from the middle of the lake. If you're swimming on the beach, it might be really different. So let me tell you more, though, about how we do this in the middle of the lake. So We have a water quality buoy. I'm assuming that many of you know that. Does anybody who has the app on their phone? So the Hoofers group and also the Space Science and Engineering Center keep very close tabs on the meteorological variables that come off this buoy. So it measures wind speed, temperature, humidity, because it affects sailing and important things like that. But below the water, we also are measuring a lot of important things like water temperature, dissolved oxygen, which for research purposes tells us a lot about the state of the lake. We only measured dissolved oxygen at one point. We don't measure it all the way through the water column, so I think fishermen would prefer if we did. But we at least measure temperature throughout the water column, which I know a lot of fishermen use to decide where they're going to, at what depth they're going to try to catch their fish. We also monitor phytoplankton pigments, so chlorophyll and phycocyanin. These are two pigments. So I would say the things that give the cyanobacteria color, right? So the green part of that, we measure with a fluorescence detector. So it actually shines a fluorescent light through the water, and then how much light actually gets absorbed and reflected back onto the sensor tells you, presumably, how much cyanobacteria is there, how many cyanobacteria. And we monitor this on timescales of minutes. So every 10 minutes it logs data and sends it back to the Center for Oceanography and then puts it into the website. So you can see it. It's a little tricky to find anything other than the water temperature and stuff online because it's hard to get those data because there's a lot of it, right? So getting it onto your computer. We measure a few other things for research purposes, but I would say that the phycocyanin and the chlorophyll are the ones that get really interesting in terms of thinking about the cyanobacteria. So this is data from 2008 because I like to show this because it's probably the best case for why we need this buoy. Okay? So this is mid-July 2008, again one of the worst bloom years that we've seen since we've been looking at them. And I'm showing the phycocyanin concentration are the blue jagged lines, and see how they're going up and down like that. So that's because of the water moving back and forth in front of the sensor. So it's not a very clean signal. It's not an easy thing to read. The black dots are kind of trying to track the average of that, so you can get some sense of the trend. Green is chlorophyll, so that's the pigment that's in algae, true algae. Cyanobacteria have it as well, but the main pigment that makes cyanobacteria look kind of bluish is the phycocyanin. So that's kind of the thing that you look for to see if the cyanobacteria are there. And my students went out and collected some samples on July 16th, because that was our regular sampling day. We go about once a week, a little more frequently than LTPR. And there wasn't a whole lot going on, so like, okay, whatever. July 18th, they were sitting on the terrace, having some beer, and one of them had an iPhone, and this is back in 2008, not an iPhone, and he was looking at the app. I think it was Lucas, actually, was looking at the app that was pulling these data off the buoy, and noticed that this was trending up. I don't know why they weren't checking it on at 3 a.m. on the 17th. So they're checking. They said, "Oh my gosh, it's going through the roof. We have to get out on the lake." So they went. Maybe I shouldn't confess this on the tape. They went to the CFL, got in a boat. They hadn't had that much beer, and went out on the lake and got a sample. And so we actually have very extensive characterization of that sample. We know that this is right around when the toxin concentrations were extremely high. And we know all about the species of cyanobacteria were there and the chemistry and blah, blah. The only reason that we got that sample was because of the buoy, right? So if we hadn't seen this on the iPhone app, then we wouldn't have sampled again until the following week and the bloom was gone. Right? So this adaptive sampling is really critical for catching the blooms, and it's not easy to do. And so I guess I would make a plug for as much automated monitoring that we could possibly afford in order to really get a handle on the transience of some of these events. And this happened to be one of the few cases recently where, recently by like the last 10 years or so, where there was actually enough press about a toxic event that it showed up on our radar where a girl went swimming and on that same evening that we collected the sample and reported cyanotoxin poisoning. It was Christine. Oh, you guys know that? Yeah, Christine. You don't know Christine. And so this, I think, was a nice convergence. I mean, I don't want anybody to get sick, but it was a nice convergence convergence of the buoy showing its value, the students being proactive, going out and getting the samples, being able to really figure out what's going on and linking to something that the public actually cares about. And so a big driver of using that buoy was Todd Miller. And so when he moved to UW-Milwaukee and got his own faculty position, he started a similar monitoring system on Lake Winnebago. So I know it's not the Yahara Lakes, but I wanted to give you a sense of what's going on elsewhere in the state related to this. So he has a very similar water quality buoy that is even cooler, so it monitors more things. It also automatically collects water for later analysis, which is something ours does not do. We have to get the students out there. And he can actually monitor all kinds of different toxins. He has his own fancy instrument that measures the toxins. If you pay to have these measurements made to the accuracy that I've been showing the numbers, it's several $100s a sample, right? So it's not cheap. He has his own instrument, so he can make as many measurements as he wants. It's a very expensive instrument, but he has one. And he has a big grant from the National Institutes of Health to study this in Winnebago. And these just show some data from 2013, so he hasn't shared his data with me for this year yet. But basically, he's also monitoring that chlorophyll and phycocyanin and the toxins. And what you should look at here is the red line is microcystin. He's also looking at anatoxin, which is another toxin that we haven't looked at much in the area. But first, you should notice that the toxins are high even when these pigments are not. So I think most people studying cyanobacterial blooms would argue that the best way to monitor them are these fluorescence sensors. And what Todd is finding is that the toxins are there even when the sensors tell you that there's not working with cyanobacteria, which was a surprise and something that certainly is troublesome because we think that the fluorescence detectors, we thought that the fluorescence detectors were about as good as you could get. I mean, beyond just actually going out and getting the sample. So these are some interesting data that are coming out of his work, and so if you want to follow up more on that, I'd encourage you to. He also partners with the drinking water utility because a lot of people that live around Lake Winnebago get their drinking water from Lake Winnebago. So that's a much more analogous situation to Toledo versus here, right? We don't use our lake water for drinking water. So this is largely why he's involved. I wanted to say a bit about this spatial heterogeneity, bring it back to Lake Mendota. So we all know that the water moves because of the wind, and the way that it moves and the rate at which it moves controls a lot of where the cyanobacterial blooms are. And again, Chinu is the master of that, so I would defer to him. But this is a very famous image that was put together by the Space Science and Engineering Center of cyanobacterial blooms. It's showing the heterogeneity. So if you go out and measure, so the buoy is around right here, so this water is moving past the buoy. If it happens to be right here, it looks like there's a really intensive bloom. If it happens to be right here, it looks like nothing's going on. So this challenge of where to measure, again, becomes important. And certainly if you're at a beach, it's different, more different. So yes?
Can you go back one slide?
Yeah.
How often do they do this type of picture? Is this done once a year? How much
Very
does it cost?
Very rarely.
How can we get more?
I would invite someone from the Space Science and Engineering Center to come and talk, and I can give you some leads on that. But this particular image was done by a researcher, and it took a lot of effort, so this is not done very often. Certainly at this level of resolution. The satellites pass over at different frequencies taken by satellite. And different satellites have different resolutions. So you need to have the bloom coincident when the satellite went over. You can't have any clouds because that obscures the satellite image. So getting that convergence of all those things happening at once can be tricky. But there are people that are interested in that at the Space Science and Engineering Center. I collaborate with a few. They're actually, I don't know if you know this, but UW-Madison is like famous for their satellite research. Like they have this big center there that brings in like tens of millions of dollars a year from NOAA and NASA to make satellite images. They are the ones that when you see hurricane images on the news, it comes from them. Right? They are massive at this stuff. So I know everybody here cares about like what's happening, where I'm swimming, or where my house is, and where my kids are swimming, or my dog. And I wanted to make that point that a lot of what I showed is, all of what I showed is measured in the middle And so Kirsty's lab does regular monitoring of the beaches. And when I asked her about this yesterday, because I was prepared for this, she pointed out to me, and this is important to remember, that for public health purposes, the measurements need to be fast, because you need to be able to get a warning to people to not come in contact with the water as soon as possible. The methods that I use to make those measurements are not fast. Okay, so they're for research purposes. So Kirsty uses these test strips that you basically put the water on the strip and it can give you an estimate of the toxic concentration. It's not, as accurate as or as sensitive as the method that I was showing, but it's fast, it's much less expensive, and you can do a lot of them. And so her group actually monitors the situation of beaches based on whether the lifeguards, trained lifeguards and other monitoring folks actually see sort of greenish tinge in the water. They take the measurement with a test strip. If it tests high, then they will do an additional test, often using a more expensive, slower, but maybe more sensitive and more accurate technique, which is still not the one that I showed you, because that is hundreds of dollars. And based on sort of that evaluation, they will decide whether or not to close the beach, right? So everybody wants to know, how is this going to affect my use of beach? So, yes?
I have a question. Could you hand those test strips out to people who live around the lake so you would have citizen monitoring?
Presumably. I actually, one thing I don't know exactly how much they cost. Do you know, Kirsty, off the top of your head?
It's about $400 a kit, and that
Yeah. Yeah, it's still not. I mean, it's an order of magnitude less than the method that we use in the lab, but it's still, you can't really go in and do that. Did you have a question?
I want to chime in that when we wrap up, we'll tell you a little preview of what CLA is doing to try to help with citizen monitoring. We'll tie in when Trina wraps up.
Yes, exactly.
Your lifeguards are tested based on the color, right?
Yeah.
And yet you're showing the toxins are higher, or can be much higher otherwise. Do they have a regular system for testing their, are you measuring toxins or are you measuring the color?
The toxins with the test strips, but there's so many beaches and there's so many days that you can't do that every day. So, correct me if I'm wrong, Kirsty, but basically you need to be notified by someone who's at the beach who notices, that it's looking kind of green to go make a point of going out there and monitoring it.
Maybe sometimes test, after the urine has been made and we usually
So to trigger the testing, you would have to be.
know how long it takes for the
Yeah, that's actually not. So and let me just sort of go through the workflow. Basically, a lifeguard or someone else would notify Chris's office that it looks kind of greenish. They go out, they collect a sample, or maybe someone else collects a sample and brings it in. Test strip tells you whether or not there's toxin there. They will then go out the next day, maybe the next day, maybe the next day to track what happens with that balloon on the beach. So, because sometimes the first test comes back low or non-detectable, but then the next day, if the balloon is starting to senesce, decay, break down, that's when the toxins get released, and so then you might see a spike in toxin levels. But we don't know how long that takes often for that spike to happen. We don't know the ways in which it gets degraded. So the toxin doesn't hang around in the water forever. Bacteria actually eat it. It's a very valuable resource for the bacteria because there's a lot of nitrogen in it. And so the bacteria will eat it up. But we don't know how long that takes. So you have to track after the event for some reason.
So I think it's similar to the question you had, but you had a slide where you can have low quantum and still not. So we really need to be testing regularly, not just when you see it.
If you can afford it. Sure.
But then,
I'm all for more data. Build me,
is there a
100
sense
more
of,
photos.
we don't have to worry because it's not clear when you really should be.
I think we don't have a good thing. But the best thing that's the best we can do, really, unless you have a budget to be monitoring it every day at every beach.
Well, even the swimmer, so she had a reaction to the article said that the weather was swimmers. So are there people who are more susceptible to it that are going to have a greater reaction than others? Others might be okay.
So the question is, are some people more or less susceptible to the toxin effects? I'd say we don't know. I don't think that kind of research can be done, actually. Yes.
With all the agencies that monitor the lake, being the most studied lake, why can't we do this basic research?
I would say, maybe, should I use the standard things? We don't have enough money. No.
Can you repeat the question?
Why can't we figure this out? Like, this is the most studied lake in the world. Why can't we do this research and why can't we figure it out? I would say that people have been studying this on our lake and in other lakes like Erie and around the world for decades, and we can't figure it out. It is a wicked hard problem because so much has to happen. You have to have the right environmental conditions for the cyanobacteria to grow, right? But just because the cyanobacteria are growing doesn't mean that there's toxin present. Not every strain of cyanobacteria can make the toxins. Some of them can, some of them can't. And we don't know why one appears and one doesn't. You might have the strain there that can make the toxin, but that day it doesn't happen to make the toxin because we don't know why they make the toxin.
More specifically, with all the agencies that are monitoring it, can't we work together and then get the monitoring since there's so much money that is coming into the studies?
You would think yes, but I think it's just a really hard problem. And it takes a lot of monitoring, which is not cheap. Like, it may seem like there's a lot of money going in, but when you're talking about $200 a sample, and multiply by that by the number of days and the number of beaches, and it probably varies within a day, so you really should be monitoring like every hour if you really don't know. And then there's the question of whether if you detect 5 parts per billion in the lake, does that mean someone's going to get sick or not? So there's a lot of uncertainty, and I know people hate it when scientists say that, but it's just the problem is that we don't understand it.
The source, whether phosphorus or nitrates, the Lake Erie farmers got the blame. It's not just farmers. And you want to just expand on that, what we can do to prevent this, all of us, because all of us live in the water somewhere in the it's, it's not just keeping water out, it's keeping out anything that we're putting on our fields, but our lawns and our...
Yes, so phosphorus and nitrogen is in fertilizer, and that's what fertilizer is. So anytime you're using fertilizer on your grass or anywhere, and that ends up getting into the lake, then yeah, that would be where the person has an impact. In Lake Erie, I would say, every time you go to the bathroom, you are putting phosphorus into the water, right? So at that level, we are all responsible for phosphorus pollution, right? It's very simple. Now, it's up to the wastewater treatment plant operators to make sure they get it out. But, we can't sit here and say it's, I don't have any role in this because we all are people who clean every day. Our sewage doesn't go to Lake Mendota, so Lake Mendota is, I think we have that pretty nailed in as far as the agriculture. But there is also some resources as well.
So your research is on Lake Mendota, but that picture showed Lake Mendota also had effluent. Do you have any sense of difference between the two lakes and the conditions? Are they pretty comparable? Has anybody been doing that research?
We have 1 year of data where we looked at Mendota, Monona, Kegonsa, and Wingo. And Kegonsa was by far the worst. And this gets at the complicatedness of this problem, that the same species of cyanobacteria are in all the lakes. So the same cyanobacteria are there, but they're making different amounts of toxin in each lake at different times. And that's because we don't understand exactly why they do it or when, such that we can't predict it. But we do know that in terms of the bloom intensity, Kegonsa in 2008 was way worse than the other ones. I don't think we have enough data on Monona or any of them longer term to know what they're like long term. But I know, Christy's lab looks at beaches on those lakes as well, so she has some understanding.
So one of the most sensitive chemical things that we all test is smell. Is that related?
Trina, can you repeat the question?
Yes. Well, I think of the answer. So can we use smell? Because humans have very sensitive noses and can tell when There's certain organic compounds produced. I mean, you can smell when they're decomposing, right? It smells bad. I don't think that you can use your nose at this point to tell if there's toxin present. I wonder if you can train a dog to smell things, right? You have monitoring dogs running around nuisance that you drink it. I don't know. I don't know if anybody has looked at that. It's a great question.
Actually, Jennifer Graham with the USGS did some work on the taste and odor compound that blooms in Kansas lakes, I think, and she showed that the presence of the taste and odor compounds like GLs when it kind of smells like damp soil, that was highly correlated with toxin production in blooms, but not always. So you can't always use that. You can't always use that smell.
Yes, there is a lot of work. I should say there is a lot of work on taste and odor in mainly drinking water treatment plants, right? Because people complain when their water tastes bad, and a lot of that can be linked back to blooms happening in the lake. Whether or not you can go to the lake and just look and tell if there's toxin there, I think there's a lot of work on that.
So we can either have you do a wrap up, pull it all together, or we can take one more question. So Dr. McMahon will be here for a few minutes after we close for more questions. So I'm going to let her wrap up, and then we want to tell you a little bit about our citizen monitoring program, which relates directly to this. So we're going to let Trina wrap up here.
I guess just to, I skipped a few slides because we were out of time, but basically what I wanted to say was that, I guess take-home messages are, yes, phosphorus is important. Keep working hard on the phosphorus. Know that that is a complicated problem, and that nitrogen seems to be important. We don't understand yet why. Monitoring is equally important. And what else is going to be? I guess I wanted to talk also a bit about how climate change interacts with some of these factors, but I've run out of time on that. So you might hear from someone else someday about that. But people always ask me at the end, would I go swimming in the lake? And I say, I need more data before I go swimming in the lake. I haven't been swimming in the lakes in a long time, I'll just say that. And I'll take all my funding sources and sponsors and stuff here. But, yeah.
How prevalent a problem is it? How many people get sick? It seems as if due to the lack of monitoring, the human being may be the best test, right, to say that. But is that tracked?
So I could go on for another 10 minutes about that, but I think that we want to let people break, so I can certainly address that. But the question was about how many people get sick and how much we know about that and whether they have a good monitoring system. But I'll let you all break.
First, please join me in giving a big thank you to Dr. McMahon. And there's a couple things I want to share. I know some of you need to head out to work, but there's a couple of important points we want to make. One is that through our whole Yahara Lakes 101 series, we have other speakers coming in to talk about these sort of side topics that Trina is mentioning. So we had Dr. John Magnuson talking about climate change. We had Dr. Emily Stanley here talking about the long-term ecological research. And what we're hearing from multiple scientists who are coming is the importance of this monitoring. Clean Lakes Alliance is working with the Center for Limnology, exploring ways to help increase the monitoring that's done through the buoys, how the community can help with that. And we're also very engaged in our Citizen Water Quality Monitoring Program, which is also working with researchers like Dr. Dick Lathrop, who's here today, in trying to help contribute data so that the scientists can learn more about under what conditions these blooms are forming and when and how that varies around the shoreline. So I'm going to turn this over briefly to James Ty to have you get out your smartphones and call up a website that we're working on for this.
So normally at these type of things, you're not supposed to get out your phone and look at your Facebook and all that type of things. I'm actually encouraging to do that right now. So everybody who has a smartphone, you get that out. If you have a real smartphone, you can actually go onto Safari, right, or whatever Chrome or whatever you use, and type in ccw.cleanlakesalliance.com. And so actually, if it's really smart, it'll just sort of come up. CCW, which stands for Clear Clean Waters .cleanlakesalliance.com. So who has that up? So I see about a couple hands going up. So this is a partnership between Dane County. I actually met with Joe Parisi and the mayor of Madison, Mayor Paul Soglin, and the University of Wisconsin-Madison. And this is the beta site. This is not even launched yet. But it's ccw.cleanlakeslions.com. And this is an app that actually will tell you what beaches are open, which ones are closed. You can click on them. And then the other thing is if you click on lakes, you'll pull up all the lakes. If you touch one of those things, if you touch like Mendota or something like that, you'll actually see our 44 citizen monitors that are on our lakes on a weekly basis, and they're doing testing about clarity and temperature and all these other things. And that's going to be used by our scientists locally to figure out what's going on. And if you actually then click on one of those things, they come up pretty cool graphs. They'll get better of what's going on the lake. And so the idea is we're going to add on the buoy. We're working with the DNR to do SecuDisc and StreamDisc. And so the idea is to bring all of the information that's either being done by government, citizens, testing that being done by the buoy, bring it all together so that citizens on a real-time basis can get as much information as possible. And then you can make your decision if you're going to go in the lake. And I'll tell you, I have a wedding this weekend, but I'll be in the lake on Sunday. No fear, I'm going in. And I'm sure my dad will be too, and Mary Lee Gorman, and a whole bunch of other people. So I swim in the lake, but maybe other people don't.
I'm going to remind you that in November, we'll have our whole 101 series. Our session will be on the results of our citizen monitoring program, more about this website and app. And we get that question a lot about, I think sometimes when you have that much information like Trina has, you look at it from a different perspective. And what we're trying to do with our citizen monitoring too is show localized how at one beach it might be pretty nasty, whereas at another beach it's crystal clear, beautiful. And so we're trying to get that information out. At some point it's going to alert people that this problem is here. On the other hand, it can be an assurance that it really is a great day to get out. You just got to look at what spot along the lake it will be. So with that, we want to give a huge thank you to all of you for coming out. We want to thank Dr. McMahon. We want to thank Foley for being the hosting sponsor. We want to thank all the Friends of Clean Lakes. Please consider becoming a friend today. We hope to see you next month. Thank you.
Right. Well, and so it occurs to me just in the past, so it probably takes a tremendous amount of that thing that's and the Wisconsin Health Department actually has a statewide monitoring
My mantra is